All right, today we're putting on our detective hats and diving into the code of life itself. We're going to be looking at pairwise sequence alignment, which is really the fundamental tool scientists use to compare the DNA and protein sequences that write the story of, well, every living thing. So let's just jump right in. Picture this. You're a scientist, you've been working hard in the lab, and you've just discovered a brand new gene. It's a string of code, right? A sequence of letters. But what on earth does it do? What's its job? This is without a doubt one of the biggest questions in all of modern biology. Well, the answer lies in comparison. To figure out your new gene, you have to check it against this massive global library of sequences we've already decoded. You can almost think of DNA and proteins as molecular fossils, these ancient texts that have been copied, tweaked, and passed down through evolution for billions of years. So by aligning your new sequence with the known ones, you can start to read its history and figure out its purpose. So how does this actually work? Well, it's all based on the simple mechanics of evolution. As life evolves, genetic sequences change and drift apart. But natural selection is a pretty tough editor. It makes sure to preserve the most important parts, those little regions that are absolutely critical for a molecule's structure and function. And this right here is the core principle of our entire investigation. When we line up two sequences and find that they look really, really similar, that's a huge clue. It's a strong suggestion that they both came from a common ancestor and probably do a similar job inside the cell. This is exactly how we can take a total mystery gene, find its closest relatives, and make a fantastic, educated guess about what it actually does. Okay, before we go any further, we have to get our vocabulary straight. In this field, there are two words that get mixed up all the time, but they mean totally different things: sequence similarity and sequence homology. And trust me, getting this right is absolutely critical. First up, we have sequence similarity. This is just a number. It's a direct measurement you get from your alignment. It's simply the percentage of positions where the letters, or residues as we call them, are a perfect match. It's a fact. It's data. You can say, hey, these two sequences have 40% similarity. Plain and simple. Sequence homology, on the other hand, is not a number. It's a conclusion. It's the scientific inference we make after we see a high level of similarity. Homology means that two sequences share a common evolutionary ancestor. It's a qualitative statement. It's an all-or-nothing deal. So here's the bottom line, the thing you absolutely have to remember. You can say two sequences have 40% similarity, but you can never, ever say they have 40% homology. It just doesn't make sense. They are either homologous, meaning they share a common ancestor, or they're not. Think of it this way. Similarity is the evidence you present in court. Homology is the final verdict. Okay, so this leads us to the million-dollar question, right? What's the cutoff? How much similarity do we need to see before we can confidently say, yep, these are homologous? And the answer? Well, it's not one magic number. It actually depends on how long the sequences are and what the odds are of a match happening just by pure random chance. This chart here gives us a really fantastic picture of the answer. It plots the length of a sequence against the percentage of identical letters, and you can see this curve, right? It shows us that for really short sequences, you need a super high percentage match to be sure it's not just a random fluke. But as the sequences get longer and longer, the amount of similarity you need to be statistically confident gets much, much lower. So scientists have kind of divided this chart into three main areas. If your alignment score falls up there in the safe zone, you can be really confident that your sequences are homologous. If it's way down in the midnight zone, the similarity is just too low to make any reliable call. And then there's that murky area in between, the twilight zone, where things get tricky. It could be a really distant relative, or it could just be a coincidence. So let's say we have a potential match. How do we actually do the comparison in the best, most rigorous way possible? Well, this is where our alignment toolkit comes in. There are two main strategies for lining up a pair of sequences. We call them global and local alignment. Think of it this way. A global alignment, which you see on the left, tries to match two sequences across their entire length, from end to end. This is the perfect tool when you're comparing two closely related genes that you expect to be similar all the way through. But a local alignment, on the right, is different. It doesn't care about the whole thing. Its only job is to hunt for the single best patch of similarity between the two sequences. This is awesome for finding a shared functional piece, like an active site, in two proteins that are otherwise pretty different. And to actually run these strategies, bioinformaticians rely on some really powerful algorithms. We're going to look at two of the foundational methods that really started it all: the simple, visual dot matrix method, and the much more powerful and rigorous dynamic programming approach. The dot matrix, or dot plot, is probably the simplest way you can visualize an alignment. You just put one sequence on the x-axis, the other on the y-axis, and you put a dot wherever the letters match up. A nice, clean diagonal line, just like you see here, means you've got a strong region of similarity. And if you see a break in that line, well, that's a clue that there's been an insertion or a deletion. But for a truly precise, mathematically guaranteed best answer, we need dynamic programming. It's this incredibly powerful method that breaks the whole problem down into 3 steps. First, you set up a grid, just like the dot plot. Second, you systematically go through and fill that grid with scores, calculating the best possible score for every single position. And finally, you find the highest score in the whole grid and trace your path backward. And that path reveals the single best, optimal alignment. And here's what it looks like in action. The algorithm just chugs along through the matrix, cell by cell, calculating a score for every possible pairing of letters. Then, it starts from the best score and follows the highest scoring path back to the beginning. As you can see, every diagonal step is a match, while a horizontal or vertical step means we had to insert a gap. That final path is the best possible alignment. It's guaranteed by the math. But wait a second, how do we get those scores? It's not as simple as saying +1 for a match, -1 for a mismatch, especially in the world of proteins. See, some amino acid substitutions are chemically pretty similar and happen all the time during evolution, while other substitutions are really drastic and rare. Our scoring system needs to reflect that biological reality. And to do that, we use what are called substitution matrices. The 2 most famous are PAM and BLOSUM. PAM matrices are based on a full-blown evolutionary model, trying to predict how sequences change over huge amounts of time. BLOSUM matrices are a little different. They're built by directly observing which substitutions actually happen in the most conserved, important parts of related proteins. And get this, the numbering is totally backward between them. A high PAM number, like PAM 250, is for distant relatives. But for BLOSUM, a low number is for distant relatives. Choosing the right one is absolutely key for your search. Okay, we have our alignment, we have our score, we've done all the heavy lifting. But there is one final question left, and it might just be the most important one of all. Is our alignment a real, meaningful discovery, or could we have gotten a score that good just by pure dumb luck? To answer that, we have to turn to statistics. It turns out if you just mash 2 completely random, unrelated sequences together and score their alignment, the scores don't just land anywhere. They actually form a very predictable statistical pattern. So by comparing our real alignment score to this random distribution, we can calculate the odds that our result was just a fluke. This comparison gives us something called a p-value, or probability value. And all you need to know is this. A very, very low p-value means your score is a true outlier. It's way out there, far away from what you'd expect just by chance. This gives us statistical confidence that our alignment reflects a real biological relationship. It's the final piece of evidence we need to close the case for homology. So we now have this amazing toolkit for comparing 2 sequences head-to-head. But in the real world, a detective doesn't just have one suspect, right? They've got a database with millions and millions of entries to search